We're going to have Khandis Blake speaking, followed by Sophie Lin. So I'll start by introducing Khandis, who is an expert on sexual politics who combines insights from evolutionary psychology, biology, and big data to understand conflict and competition among people. Her research addresses big issues that profoundly influence well-being, including personal agency and empowerment, intimate partner violence, and the varied ways in which people seek and enact status. And today, Khandis is going to be talking to us about some work exploring psychological patterns across time using 2.3 billion tweets. So I'll hand over to Khandis.
Thank you. All right. Thank you, everybody, for coming today. I'm delighted to be able to talk to you about this work. Elise did a great introduction, and that is indeed exactly the kind of things that I do and the work that I research. So I am in the Ethics and Wellbeing Hub, and I'm the director of the Evolution Lab. And just like Elise said, what we are really interested in is sexual politics, broadly defined. And what we tend to do is take very ideologically divisive, complex topics like attitudes towards intimate partner violence or gender roles or biological indicators of behaviour and personality like menstrual cycle hormones, and try to understand these topics by using mainly biological or evolutionary models. So this is some of the work that we've been doing here. And to do this, we look at micro, meso and macro processes, and we try and take a nature-nurture view. So my background is in social and gender psychology, but also in evolutionary biology. So our work is quite interdisciplinary. So to give you a brief background, in terms of the micro-level effects, here what we tend to do is look at individual variation in behaviour and cognition and attitudes across the menstrual cycle and as a function of menstrual cycle hormones. And this work involves a platform I've set up called the Daily Cycle Diary, which is a big data platform where anyone in the world can come and log data on their menstrual cycle and also answer questions about their psychology and behaviour. Now, my lab is rather quantitative. We get quite into being technical and doing complex statistical things. And what we've done is code this data so everybody gets a free, personalised 12-page report on how their cycle affects them or doesn't affect them. And they can use these data to try and understand patterns of their own behaviour, but then to compare themselves to all the other women around the world who've done the survey, depending on whether those women are on birth control or naturally cycling. And we've had 1,200 women from 25 countries do this. And let me just get the mouse across here to give you an example. Okay, the report is great. Okay, you're all really impressed with it. It's 12 pages. It shows them all these graphs on their cycle and their data, etc., etc. Okay, so where was I going with this? Here, right, so okay, so right now we're using these data to understand health across the menstrual cycle. So we have this idea that female health in its totality is actually cyclical, and it's got a lot to do with menstrual cycle hormones and the cyclical nature of the menstrual cycle. So this is the kind of stuff we're doing right now, and we're looking at this work also as a function of hormonal birth control. Now, in terms of this level of work, what we do here is examine how family and kinship structure and close relationship contexts can operate as sites of gender norms and as sites of negotiations of power. So we look at things like the number of close kin you might have and the sex composition of that family network, and whether that sex composition could predict your political attitudes. And for those of you who are biologically minded, the idea here is that some of the political attitudes we hold are actually very self-serving, but they may not serve your individual interests. They may serve the interests of your kin network as a whole. In other words, your inclusive fitness. Now, in newer work with Yoshi Kashima and Saeed Shafa here at the school, and also with Wen Chen, what we've been interested in understanding is how kinship practices concerning marriage and lineage can be used to understand the cultural evolution of gendered norms. Okay, so now onto the macro level work. This is the most relevant for the talk today. And here what we do is try to understand ecological characteristics and how they affect sexual politics and sex and gender differences. So some of these characteristics are things listed here on the left. And we do this kind of work in two ways. We manipulate these things on the left in an experimental context, and then we measure these things on the right. But the other work we do is we then measure these things on the left, kind of like they do in sociology, and correlate them with these things on the right. And it was this work that led me to create TwitPlat, which is the platform I'm going to talk to you about today. So what is TwitPlat? Well, it's a database of 2.3 billion tweets, 10 years of data, and it's time-stamped to the second. It came from various sources, mainly Twitter's sprinkler stream. And what sets it apart is not only the scale and quantity of the data, but also the geolocation. So this data is geolocated to every UN recognised country, every city in the world with a population greater than 100,000, every city in the US with a population greater than 5,000, every state or region or territory of the top 10 countries with the most Twitter users, and it is freely available for research purposes. So where did it come from? So this is something I developed during my postdoc in evolutionary biology. Live streaming Twitter data was possible then, not possible now. And what I did was set up a series of computers that would stream the data live and then geolocate it and archive the data. This was all pre-Musk and pre the X takeover. And here's the log on here. So if you want to take a copy of this, you're welcome. You've got a laptop, you want to kind of fiddle along as you go along, please feel free to do that. And if you want a personalised login to you, let me know at the end and I can create one for you. Okay, so why did I do this? When I finished my PhD, I really wanted to do some big data stuff. I kind of wanted to do that in the PhD, but it didn't happen. Just thought big data seemed really cool. So when I started my postdoc and had some freedom to choose what I was going to do, I thought I would do something looking at sexy selfies, which from my interests are this indicator of female-female competition. So I went ahead and did that, but it turned into be a much bigger game of problem solving than I realised. And along the way, that's how I developed the geolocation algorithm. And I developed it in such a way that it easily links to ISO 3 country codes and US city codes, so you can correlate it with other socio-structural data. I learned a lot along the way, and I ended up with this huge bulk of data and wanted to then find out a way to make it searchable. Now to do that, I joined forces with the Melbourne Data Analytic Program, which is an arm of the university that offers these kinds of technical services. And we got these data into a program called, well, we got this data into Elasticsearch, which is basically like Google, but for your own data. It allows you to search things very, very fast, which was really useful because here we had almost 6 terabytes of data, which is a lot. And you can do all sorts of cool search things. So let me show you what TwitPlat looks like, and just bear with me while I figure out how to get this where I want it to go. All right, much better. Okay, so this looks rather big, but bear with me. So this is TwitPlat. You could think of it basically as just, so complicated. Having a bit of trouble scrolling, but think of it as a big Excel file, right? Lots and lots of rows like this. Each tweet is a row, and each tweet has 150 variables. Now, within TwitPlat, there is a search that you can do, a general public search. So we set up this dashboard, and this dashboard has all sorts of information about how to search TwitPlat. And then it tells you information about all the tweets that we've got in there, the breakdown by year, the geolocated tweets, sentiment, etc., etc. So, okay. So, now each tweet comes with a location text, and what the algorithm that I designed does is read in that location text and match it to a dictionary of regular expressions. So these are basically just like a search pattern. And those expressions are the names of the locations that you want to match to. The algorithm determines duplicate matches and ambiguous matches and makes a decision. And what you've ended up with after running this is the 2.3 billion tweets in TwitPlat. Now, we have done a lot of different things with this. There are so many opportunities. Most of what I've done so far involves aggregate data and sociological style analyses. And what we've done is correlate ecological characteristics with these aggregate behavioural and psychological outcomes. For example, we've looked at things like sexy selfies as a function of income inequality or gender inequality in a particular area, online misogyny and how that correlates with offline, real-world crime behaviour, incel activity online and how that is correlated with the number of single women in an area or manufacturing shocks. What I'm going to do today is tell you a workflow for how you could do that kind of analysis, and then at the end I'll explain other opportunities available as well. Okay, so the aggregate level analysis follows a very similar workflow which I'll run through. You start off by defining what are you interested in, okay? What's your phenomenon and how do you get that into language? So in the previous examples we were interested in sexy selfies because it reflected female intrasexual competition, etc, etc. And basically what you want to do here is find terms that you're really confident represent the kind of topic that you care about. You do this by looking online, doing some good research in the area. You could use hashtag finders as well, which will pull up a bunch of different hashtags for you that represent your topic. After that, you need to validate your terms. And what we usually do is create a dictionary of our terms and really only use the ones that we're very confident represent the phenomena that we're interested in. And you need to remember here that mentioning doesn't equal sentiment, right? So, you know, if you're interested in something like, I'm going to give all gender examples because that's what I do, but something like International Women's Day, that doesn't automatically tell you that someone is pro or anti-International Women's Day. So you need to think this through and really check out what these terms represent in language. Try to keep the terms as simple as possible, but also keep, remember the maxim of garbage in, garbage out. If you feed garbage terms into Twitplat, you will get garbage data out. So this is a really critical step. Okay, yeah, that's these terms in a way that you can read them. Okay, so after that, what you're going to do is filter Twitplat to retain only the posts that match your terms of interest. And you do this simply by creating a search string of the kinds of things that you care about, and then what you can do is go to our download page, create the search and download the result. So, okay, download page is up here. I'm probably, maybe I already ran this already. Okay, so I think I did run this. So we run the download and what it gives you is the number of tweets representing a particular topic split by here you've got country level codes and year. Depending on your search term there might be a lot or there might be not so many. You can just go down to the end and if you click download you'll get a copy, you'll get a CSV which just looks exactly like this table. Now, after you've got that CSV, you want to correlate that with your ecological characteristics. So it could be something socio-structural, it could be something else. We have used things in the past from the UN, from the World Bank, from census bureaus, things like sex ratios, degrees of income or gender inequality, population size, median income, crimes, etc. And the way that the Twitplat data is exported is it gives you the codes that you can easily match to you at the US Census and the World Bank and the UN, which are those ISO 3 country codes or the US city codes. Okay, so other tips and tricks is there's a particular way you want to analyse these data. Okay, so these are count data, there's certain things you want to do. I'm not going to go through all of these things, but I just put this up as a disclaimer. You want to pay attention. This is not a standard linear model you've got to do. You've got to use a count model, basically. If you have more questions about that, please ask me. Now, you might want to do more complex things. So what I did was explain how you can do a kind of aggregate level analysis where you're correlating the total number of posts with some kind of structure of the environment. But you might want to do something else. So these kinds of things are available as well. You could do a linguistic analysis. So you might care about something like how does the sentiment of a topic change over time and over place? For example, do linguistic identifiers of depression correlate with season? You might be interested in the trajectory of something. So how does the frequency of a topic change the frequency of a topic change over time and place? Has stigma towards mental illness changed over time? And if so, has it changed over time more in certain places than other places? And what determines that change? Perhaps you're interested in something that's a bit more like an epidemiological analysis. You know, where and when did discussion of this particular topic or this ideology originate? How has it changed across time? How has it changed across place? Or perhaps something to do with group identity. How does a person's group identity change their sentiment towards a topic? For example, do political identities predict how people talk about something like immigration? Or maybe you're interested in more ecological shock style analysis. So how does the frequency or sentiment of a topic change depending on an external event? One thing we talk about in the sex ratio world is the loss of a major sporting loss or a big sporting event where an influx of people arrive in a particular city. So all of these things are available as well. Now, okay, so if you want to do all of these things are available and they require access to the raw data. Now, if you want to do something that is a bit more complicated, I first need to explain to you how does this data work, because the data in TwitPlat is the result of a licensing agreement between myself and Twitter, and there are certain restrictions to that agreement. So anyone can go and download aggregates. You could use the login that I just gave you then, or get a permanent personalised login, and you could download aggregates representing a topic today and go and use that for whatever you want. So anyone can do that. That's completely publicly available. But if you want the raw post for something more complex, it's available, but there's certain steps we would need to go through. You'd need to enter into a legal agreement with me, which involves you need to read through some terms and conditions and agree that you will use the data according to those stipulated terms and conditions. You'd need to be added to our ethics agreement, and we'd need to set you up to access the data in a secure research environment, which means creating a login for you to access the data yourself. So all of that is possible. It's just a little bit more complicated. So if you're in that boat, please come and see me and let's talk. Okay, so that's it from me. I hope it's been worthwhile to you. There was a few more kind of engaging things I was going to do with showing you around TwitPlat, but the technological interaction between me and the computer got a little complex, so I haven't gone through them all. So if you also want to see some of those things at another point, please let me know, and I'm happy to show you myself. Thank you for your time and for your attention.
We have about 10 minutes for questions.
Yeah, Marta.
Thank you. That's really interesting. I was just wondering, with the keywords, and you mentioned some example of not necessarily knowing the sentiment, but it wasn't clear to me how do you go about making sure that the sentiment is accurate? Because some of the questions that you had were actually quite specific.
Yep.
Without having to go through all of them.
Yep.
How do we ensure that you're extracting the correct meaning?
Yes. So this very much depends on what your topic is, like what the thing that you want to look at is. So to get there's certain things that I found I've been able to use this data for, and certain things that I haven't. So one of the things was actually International Women's Day. Early on, myself and Morgana Lizzo Wilson, who's up at UQ in social psychology, were really interested in looking at sentiment towards International Women's Day. And we tried in so many ways to narrow down International Women's Day terms to either include sentiment, so it was embedded in the term. So the term doesn't just have to be a single word, it could be a string. So it could be, I love International Women's Day, that kind of example. Or find, say, hashtags that had the sentiment embedded in it, or were only used in a particular negative or positive sentiment context. And for International Women's Day, we actually found we just couldn't do it. We tried for ages to find the right terms, but people were talking about it in such a varied way and sarcastically, and it just wasn't possible. But in other work, like say the incel work here, right? So these are all terms from the incel movement, which stands for involuntarily celibate movement. It's essentially kind of like a spin-off of the men's rights activism movement, if those of you who may know about that. It's a group of largely young Western men who are very unhappy with their inability to have romantic relationships with women, very misogynistic, very angry, etc. And there's this big movement of these guys online. So they have a very distinct language, and they're very interested in showing their group identity through their language. So there we were able to use those terms to try and show the frequency of incel ideology, because they were the only ones really using a lot of this language. Now, it's not perfect, but that's really the process of validating the terms, is going through and kind of really making sure that you're confident that the terms represent what you want. Another thing you can do is, if you wanted to download all of the raw tweets, is you could then, that match, say, something like International Women's Day, you could then run a sentiment analysis on it. The last time I tried to do that was close to 10 years ago, actually. I haven't tried recently. And at that point, I found that the sentiment tools available for a tweet, which is 256 characters or something, very short, I didn't think were very reliable. The data, it gives you data, right? It can spit out numbers. But when you look at those, they just didn't really pass the sniff test for me of what I wanted. So I think there's probably a lot that's changed in that world since then. So I suppose my overall answer is interrogate the data, make sure you're doing what you want. It's never going to be perfect, but you might have an external kind of quantitative system where you download, say, 100 tweets and you're like, if 80 or more of them match what I want, that's my cutoff. Or you might be more involved in that. It's kind of up to you. Yes.
Do you have any annotations around demographics of who wrote the tweet?
Yes, so look, Critter is a skewed sample. This is something we know. There's a lot of work that has been published on this, I believe, from memory. So this was 2012 to 2021 is the sample. So I believe that it's skewed slightly more left. Actually don't remember more about it than that, but if you do a quick Google, you'll find the ways that it's skewed.
Is that annotated at the tweet level? So you have, for example, username Gustav, I assume it's a man.
Yes,
like
okay, so what do we have at the tweet level? So not a lot in answer to your question. So you end up with okay, so I'll yeah, okay, we'll just use the first one, right? So everybody has a username, there's the text of the tweet, there's all the geolocation stuff, there's a user description, which is static, so this doesn't vary per tweet, this is just how they describe themselves. They also can put in their actual name, but none of this is verified, right? So it might not be their actual name. So, you know, you might have your username or your screen name, Psychology Rocks, yeah? And then your actual name could be your name. So that's the rest of the information is not really identifiable to work out a kind of demographic for the user. We have developed a genderizing algorithm that reads in those first names and then gives a probability of whether someone is male or female, non-binary, or usually a company or a brand. So we've done that work. We did some machine learning work to try and figure that out. It's not as accurate as I would like, or to update, it is accurate, but it excludes then quite a big volume of the data. So it's possible to try and use tools like that to get at something like gender. If you are wanting to understand something like social economic status or age, that's possible as well, but you would be doing that by using people's language within the tweets. So it's not an external identifier of that kind of data, which itself is, you know, it's something, but it's got a limitation to it as well. Yes.
Sorry, Khandis, I think I misunderstood. I thought you were saying that you can't get the text from the tweets without going through all of those hoops of being added and entering into the agreement and stuff. Is that correct?
That's right.
That is correct. Okay,
Yep.
So what you just showed us was if someone had engaged in that.
Yes, you mean that table before? Yes, sorry, I should have specified that. So I can see different things to you because I own the system. So I was going to show you the fancy table so you can understand that it looks like an Excel table, because one of the things I find when I talk to people who commonly do individual level analyses is translating that individual level data to then understand the output of the aggregate level data. It's just a different kind of analysis. So I was going to use that as a visual, and then I had the breakdown with Excel, couldn't get the CSV file to work, can't get the table to scroll. So yes, I can see this, but the general user who logs in with a personalised login can't see that. Entering into a data agreement with me can see it. Yeah? Yes?
What does this look like post-text takeover? Is it just impossible to get updated data?
You can, you just need to pay $40,000 a year or something crazy. Yeah. So, I haven't looked into it. Other than that, they cancelled most of the academic applications. Not this one, luckily. But yeah, very expensive, and I think the quality of the data has just really gone downhill. Yes?
A cheeky question.
Yes, please. Go for it.
They cancelled so much. Why didn't they cancel you? How did you get around it? Were you
I don't
on the
know.
radar, or?
I have no idea. Don't tell anyone. They just didn't. I don't know if they just didn't cancel certain applications, but all of my API keys and my tools and all of my login, my credentials all still work. I never got any kind of cancellation from Twitter. So, yeah, who knows? But we're good to go until right now anyway. Who knows what will happen in the future? But right now, get in while it's hot, ladies and gentlemen. The data is available.
I feel like that's a good note to
Yes,
end.
okay. Thank you, everybody.
Great. Okay, a little bit of a gear shift. Our second speaker today is Sophie Lin. So Sophie's research focuses on non-invasive brain imaging, particularly using optically pumped magnetometer-based magnetoencephalography, OPMEG, for those of us who don't want to say that ever again, to investigate cerebellar function in human learning and cognition. She has contributed to developing OPMEG as a method for recording cerebellar activity in humans. Her work also explores predictive processing in both the cerebrum and cerebellum, as well as neural mechanisms underlying neurological conditions such as MS, aiming to inform treatments like brain-computer interfaces. And so Sophie is going to tell us today about the OPMEG, and I'll hand over to her.
Yeah, thank you very much. Right. Thank you, everybody, and it's good to be here. So I basically changed the title of the talk a little bit to be cerebellar magnetoencephalography. Basically, two keywords which might not be familiar to all the audience here. But so the key information I want to convey here to you is actually very simple. So the first thing is, I want to say that the human cerebellum is understudied, especially in domain of functional neuroimaging, and we need to address that. And for me, as a MAG researcher, I do have to admit that conventional MAG is basically has its limitations because of its need for liquid helium systems. But so the introductions of wearable MAG based on optically pumped magnetometers, OPMs, so can potentially open the door to cerebellar studies. And I basically want to show you, use two small studies that we conducted. But before that, so a little bit data. So on the left, this recent TICS papers, so published this year. So basically it looks at all the functional MRI papers from 2020 to 2024. And then what it discovered is papers that mention the neocortex are basically 8 times more than papers that mention the cerebellum. And it also gives the notation saying that a lot of the time when a functional MRI papers mention it's a whole brain study, actually it either only cover part or totally eliminate, not really caring about the coverage of the cerebellum. And as for MAG study, so there is a review paper published in 2020, basically look at all the MAG papers that have published and discovered around 25 studies that have mentioned cerebellar activities, and most of them serendipitous. So that's actually much smaller, much fewer than 1% of all the MAC studies that have ever published. And as for the cousins of MAC, the EEG, so the 1020 system, whether it's standardized 32 electrodes or high density arrays, don't normally cover the cerebellum either. So when I say that the human cerebellum is ignored in neuroimaging, it is no exaggeration. But you might be saying, why should we care? Maybe there are few publications in the cerebellum because, well, the origins of the cerebellum in Latin is the little brain. So it's a little brain, and it makes sense that it gets little, attention. But what I want to argue, in fact, 80%, more than 80% of the neurons in the brain were actually situated, located in the cerebellum. And unlike the conventional sentiment saying that the cerebellum is just a motor organ, that's not true. Actually, most of the functions that has been discovered in the cerebral cortex also exist in the cerebellum. And one thing I consider which is very important is the microcircuits in the cerebral cortex is actually very different from the microcircuits in the neocortex, which means when you see a copy of the function that's being discovered in the neocortex, it's not very likely the cerebellum is just using exactly the same computation to copy that function. So we have to understand what exactly it is doing and just look at the practical side. So the cerebellum in recent years has been implicated in many different kinds of neurological and neuropsychiatric disorders from ASD, psychosis, Parkinson's disease, and Alzheimer's disease, to name a few. So keep ignoring the cerebellum in neuroimaging study or in neuroscience is really doing harm to ourselves. But even to me, as a researcher who is studying the cerebellum using MAC, I do have to admit, cerebellar MAC is just hard. So we can start from the anatomical constraints. When you have a patch of neurons that being activated in the cerebellum, because of this nature, it's densely foliated cerebellar cortex, it would have a higher degree of signal cancellations compared to the cerebral cortex. That means you get smaller signals. And because the cerebellum is deeper compared to the cerebral cortex, so when that signal travels from the surface of the cerebellum to the surface of the head, the small signal gets even smaller, and the conventional instrumentations of the MAC is not really doing us any favor because, well, the conventional MAC systems using very amazing superconductors, it's very sensitive, it's really good, but in the meantime, when it operates, it needs very cold liquid helium. So that means you are separating your senses from the brain by about 3 centimeters to begin with. So you're really jeopardizing yourself if you want to look at the signal which is already very small. Yeah, and actually, similar to the situations in functional MRI, if you look at conventional systems, a lot of time, it simply doesn't really cover the cerebellum. So, we were very excited. It's really about the introduction, the very difficult to pronounce so-called optically pumped magnetometers, so-called wearable MEG. So what it really does is, instead of using superconductors, it uses a totally different physics phenomenon to detect very tiny neuromagnetic fields. So to put it simply, it uses the properties of alkaline atoms. When they are in its gas form, it would be something like a very small magnetic sensors. And what it does is it can track very small neuromagnetic field by tracking that as a way to track the laser intensity. So the physics phenomenon itself is very complicated. but you can basically manage it in a way to a very beautiful, simple linear equation that you can measure brain activity using this type of sensors. And the most important thing is these sensors, they are operative at room temperature, sometimes slightly higher than room temperature. So that means you don't have to worry about just to place those sensors directly on top of the scalp. You can get sensor closer. And another amazing thing is these sensors, they are now small enough, which means you can arrange your sensors in a way to best suit your research need. For example, for us as cerebellar researcher, we can basically arrange the sensor array in a way that can best detect signals from the cerebellum. So, back in time when I was in London, that was like 2018, quite some time ago. Back then, each sensor was about the size of a white ball marker. And when you try to place the sensors on the surface of the head, you basically need a customized helmet that's produced based on individual scalp MRI structure. And for each helmet, at that time, you can place about 100 sensors, but back then we only have about 25 sensors. So we did a little bit modeling, try to identify around 20 sensors that are closest to the cerebellum. And we basically show that compared to the Swiss systems in London, so we can effectively reduce the sensor to cerebellum distance by about a third. And because this is an exploratory study, so we want to use some stimuli, some kind of stimulus that is really powerful. And what we decided to use is basically unexpected air puffs being delivered to somebody's surrounding, really close to somebody's eyes, because there were numerous animal research showed that this is a really powerful cerebellar activator. So that's what we did. But well, the air puff is not only activate the cerebellum, it is also somatosensory stimulus, so it will also activate the contralateral somatosensory cortex. It causes people to blink, so there will be Eyeblink artifacts. So the way how we design the sensor arrays with limited number of sensors is we basically try to concentrate enough number of sensors in posterior cranium, and then we place a couple of sensors on the contralateral somatosensory area, contralateral to our stimuli, and then a couple of sensors surrounding the eye to pick up eye movements. And, well, the customized helmets means we can't really perform studies on too many people. So we did studies across 3 individuals. And overall, so across all 3 individuals, in the posterior sensors, which is the upper row, so we found beautiful multiphasic evoked responses. And there are mainly 2 peaks. For the early peaks, it centers at around 50 milliseconds per stimulus, and for the late peaks, center around 85 to 150 milliseconds per stimulus. We also discovered somatosensory and blink responses, but both the spatial and temporal features, they don't look the same as the posterior sensors. And of course, we do want to know where the signals comes from. So what we did is a really basic Bayesian modeling. We start with 6 different prior models coming from bilateral somatosensory cortex, bilateral cerebellum, bilateral medial parietal frontal areas. And basically, we try to compare model fits to data to see which models can explain our MEG data best. So we're happy to say that for both early and late peaks, we discovered the ipsilateral cerebellum was always the best fit for all 3 participants, and that's incompatible with, it's compatible, with what has been discovered in animal literature. And also, when you look at the locations of the dipole phase, so for all 3 participants across early and late peaks, they always converge to the cerebellum. And most importantly is, even when you start the prior locations from somewhere which is not the cerebellum, for example, like the median parietal occipital region that I mentioned, when the models converge, they always converge onto the cerebellum. And because the cerebellum is covered by a thick layer of muscle, so we also try to start with muscle. And again, we show that when the model converge, they still converge to the cerebellum. So we can say we have a relatively high confidence to say that we basically verified that we can recall cerebellar evoked fields using wearable MEG. And we can also say that we demonstrated that with on-scaled customized sensor arrays where MEG, wearable MEG has the potential for cerebellar functional neuroimaging. But we do want to know a little bit more about what exactly is the nature of the neurogenerators of POF-related response. So we basically did something else. So that air POF responses, when you pair that POF with conditional stimuli, which always proceed to the POF with a certain fixed interval, over time, it is very, very understandable. People learn the temporal associations, and what's going to happen in behaviors is they're going to start blinks right immediately before or right when the POFs arrives. So that's just classic conditioning learning. So the important thing is two things. One is for both animal invasive physiology and human lesion studies, we know the learning happens in the cerebellum. That's one thing. And then the second thing is from animal invasive literature. So we actually know what happened in the cerebellar cortex is. So before the learning, you have a really strong response to unexpected POF in the cerebellar cortex. And what happened is after the learning, that response to unexpected air POF, unconditioned stimulus, I would say, basically decrease. And then there would be a newly acquired cortical response now happen between the conditional and unconditional stimuli interval. So we decided to run an Eyeblink conditioning OPMEC study using very similar paradigm and to see if we can discover similar changes. So we basically repeat the studies with 4 individuals. And for the baseline, for the unexpected air POF, we basically repeatedly discover beautiful multiphasic evoked response post unconditioned stimuli, and the source localizations again identify the source to be in the ipsilateral cerebellum. We then introduce about 700 trials, allow people to learn the temporal associations between conditioned and unconditioned trials. So one thing is behaviorally, they did start developing conditioned blinks, and then what we discovered is indeed the evoked response post POFs now reduce just as what the animal literature discovered. I didn't show it here, but what I also want to tell you is that reduction has no differences whether they are people start existing condition. Well, I should say when we separate the trials to trials with condition and without unconditioning trials. So that means when they learn the association, this type of reductions of evoked response is not actually conditioning to the blink behaviors itself. That's the first thing. The second thing is, unfortunately, at this stage of analysis, we didn't discover that newly acquired evoked responses or the newly acquired cerebellar cortical responses between the conditioned and unconditioned stimulus. So we did a further analysis. There's basically literature saying that the newly acquired cortical responses, cerebellar cortical responses, can be seen to be highly correlated with conditioning blink. So what we did is we basically re-evoked all these MAC responses to the onset of conditioning blinks, and indeed we discovered a small but significant evoke responses just prior to the peak of the conditioned blink in 3 out of, not all, but in 3 out of 4 participants. And again, our source localization identified ipsilateral cerebellum to be the source. So, a short summary is, overall, the MAC findings in the blink conditioning paradigm largely, generally align with the invasive animal electrophysiological finding, and we show the potentials of wearable MEG to bridge animal and human research on cerebellar function. And hopefully, by this time, you will kind of agree with me that first thing is the cerebellum, the human cerebellum, is under studies and need more investigation. And the second thing is the use of the wearable MEG open new opportunities to cerebellar imaging studies. So if you're interested to work with me, talk to me. And that's the end of the talk today, my talk today. So I want to say thank you to all those amazing former colleagues back in London, back in Britain, and also want to thank you for your attention.
Yeah, thanks for a fantastic talk. Really cool stuff. I just had one question about the sources of these signals coming from the cerebellum. So, in EEG, the sort of conventional wisdom is all these aligned pyramidal neurons which create this beautiful, nice electric field. Within the cerebellum, you did mention that the anatomical organization, the cell types or maybe even the layer structure is quite different. Do you think that you're picking up on the electrical or the magnetic fields generated by certain types of cells within that? Or is there anything about the orientation or the structure of certain cells that you think might be more dominantly contributing to the MEG signal? Yeah,
that's a really good question. So actually, for the cerebellar cortex, the only type of the, I would say, neurons, the main neurons, and also the main neurons with effective inputs and output, that's the Purkinje cells. And also, fortunate for us as researchers, they very much like the cortex, align very well, like in a cortical sheet. So there were some modeling work showing that's the most likely sources for non-invasive electrophysiological studies to pick up effective signals. So that's one thing. There were some small disagreements because there are some other types of interneurons, just like the cortex as well. But I think at least we can say we have high confidence based on both the modeling, and also so some animal study pick it up by MAC sensors when we actually isolate animal brains outside their hair to say we can pick it up. We have high confidence to pick it up signals from the Purkinje cells, but it's something of ongoing debates whether we can pick up signals from other cells as well. Yeah.
No, sounds lucky for us.
Yeah. Hiya.
I'm curious about your Eyeblink conditioning results.
Yeah.
Would you want to bring those back up on the...
Yeah, sure. Oh. No. Not happy. Sorry, is it moving? Yeah.
This one, yeah.
Yeah, this one?
Yeah, so I'm curious about the finding you expect that you have on the left is that with learning the response to the unconditioned stimulus sort of transfers to
conditioned stimulus.
Mm-hmm.
What do you think it tells us that that's not being shown here? Is
So,
it that a different algorithm is it happening at the level of the computation, or is it something to do with the measurement in the imaging?
yeah. So in that paper it just got accepted. We have several hypotheses. So one thing I do want to say is actually, so out of all 4 participants, we did actually see one existing that newly acquired evil response between the conditioned and unconditioned stimulus interval. So probably a couple of things. The first thing is we did this study in a very, constrained environment back then. So we probably didn't optimize the conditioned learning paradigm. That's based on we have moderate of like conditioned behaviors. So that's something we're thinking. Maybe that might be a reflections like incomplete learning. That's one thing. The second thing is we do need to admit for this one, that's basically invasive like recordings. So it's more like you try to extrapolate the finding from relatively small number of neurons to when you are recording with a big group of neurons. And we focus on the Purkinje cells, but they are other types of cells, and also we focus on one type of firing patterns in the Purkinje cells, but they are also different types of firings. That's been going to be captured when you record things extracranially. So all these things could be putting together, making the direct inference from intracranial recordings to extracranial recording a bit complicated. Yeah.
Quick follow-up then, if I may. Does that imply that there's sort of an unknown amount of dark matter neural firing that the MEG is just not picking up? Does that make it hard to interpret null effects like these ones? If there's always the possibility that there's extra firing that you're not picking up that might support the signal that you're not
I think that is something like it will always be a challenge whenever it's irrespective of the structures that you are looking at. There will always be something to be a challenge when you try to compare animal versus human studies. So, and I can think of a couple of things in order to better address that issues, including like instead of looking at single cell, looking at multiple cell arrays, and there are also other ways like they were also Max study previously have done like concurrent intracranial versus extracranial recordings. That's like a lot of things we can discuss about, but I do agree there's always that limitations. Yeah.
If there's no more questions, I'll say thanks again to Sophie, and thanks for everyone for coming out. There's a little more food if you didn't get to eat.